All right, here's part 2 of Key Knowledge 1, the plasma membrane for VCE biology. And so far we've looked at the plasma membrane, how it works, what hydrophilic, hydrophobic mean, and some passive transport. Let's move into active transport now. Okay, probably the most important thing to remember about active transport is that it requires energy. So each of these processes, active transport, endocytosis, and exocytosis, they all require energy. Okay, so the first one is active transport. Now, this is again moving molecules across a plasma membrane, but it's different from diffusion or even facilitated diffusion because, yes, it requires some sort of protein to move things across. It also requires energy, and the reason it requires energy is because we're moving against the concentration gradient. We're trying to get water to run uphill. Okay, so we've got to draw this as well. So obviously we're going to need a semi-permeable membrane. Okay, so there's our plasma membrane. Now let's draw our molecules. Right, so there's our molecules. We can see I've got a definite difference in concentration. I've got a lot over here, so that's a high concentration, and this is a low concentration. Normally you'd expect the molecules to go from there to there, but that's not going to happen. They're actually going to move where there's already more of them. So we want to try and get that molecule to move that way. It's very, very unlikely to happen spontaneously. So it's going to need a couple of things to help. It's going to need a transport protein and some energy. Okay, there's our transport protein in purple. So we've got a gateway to let the molecules through. Now we need a little bit of energy. And what better way to draw a little bit of energy than, a little bit of a spark. We might call that ATP. Just having that energy molecule there. The protein will take advantage of it and use it to help transport that molecule against the concentration gradient. Okay, the next two, endo and exocytosis, a similar process, and they almost look like each other in reverse. The way to help you remember is endo means we're taking it in, and exo means we're pushing it out, and cytosis because the cell's kind of moving to allow this to happen. The energy is a little bit harder to draw, and we won't really include it in the diagram because what's actually happening is the energy is used to manipulate the cytoskeleton, which is kind of hard to represent in a Year 12 biology diagram. Okay, so I've popped the definition for endocytosis and exocytosis together so you can compare and contrast them. So endocytosis is a process in which a cell takes in materials from outside the cell by engulfing them and fusing them with the plasma membrane. Whereas exocytosis is when the contents of a cell vacuole, remember that's a little bubble within its own little membrane, is released to the exterior through fusion of the vacuole membrane with the cell membrane. And I'll draw them both up and you'll see how they're the same but opposite. All right, let's start with exocytosis. Okay, the best way to do this is perhaps to show it in 2 or 3 stages. So the first stage you might have your cell membrane and a vacuole. And of course you have to remember to label these so that an assessor knows what they're looking at. Another good label to include on this is the inside and the outside of the cell. So you're demonstrating that you know which way the material is moving. You know that it's moving from inside to outside the cell. And you also want to show that there's some sort of molecule or contents inside the vacuole. So that's step 1. The next step we want to show is that the vacuole joins up with the cell membrane. Okay, and here we have our vacuole fusing with our cell membrane and the molecules leaving outside. And of course, to show that, just add a couple of extra labels. There's no need to make your drawings too complex, just simple lines and clear labels are all you need to do to show your understanding. All right, endocytosis looks like pretty much the reverse. Let's draw an example of that. Okay, for the endocytosis example, I've drawn a line with this funny green blob on the outside, which probably doesn't mean a lot because I haven't done my labels. So obviously this, hopefully you can realize, is a cell membrane. This is outside, and this little green shape is a bacteria. So I'm going to show an example of Phagocytus, which is when a cell engulfs a whole other cell. Don't forget there's also pinocytis, which is when it takes in water. Okay, let's show what that looks like. Okay, and here's the second step. So we can see that the membrane has sort of folded to surround the bacteria. So that's what's happening there. I'll add a couple of labels to explain that. And I've sort of missed the last step where that bacteria would then become part of a vacuole, which then a lysosome might merge with and digest it, remembering that lysosomes are full of digestive enzymes. So I'll just squeeze it in here by showing a little vacuole with the bacteria inside. And that's active transport. Okay, I already briefly talked about this, but there are 3 types of endocytosis. 2 of them I expect you to be pretty familiar with for the exam. And what molecules do they take into the cells? And then finally, why are vesicles able to fuse with the plasma membrane? All right, so the 3 types of endocytosis is, we sort of talked about the general one, but there's 3 special types, one being pinocytosis, which takes in fluids, phagocytosis, which takes in cells, usually bacteria, and it's usually done by white blood cells, and a special one called cell-mediated endocytosis, and that's activated by receptors on the plasma membrane, which tells it to gobble up whatever's there. So I'll put quick definitions of those there for you. I just want to make a further note on 3, the cell-mediated endocytosis, and how it takes up specific materials. All you have to do is trick a cell into engulfing around it, and that's how viruses work. That's how they get into the cell, and that's sort of how scientists are trying to stop the coronavirus from getting into our cell. Because they removed the spike protein, it can't trick the cell into engulfing it and taking it into the cell and then going through the whole replication process and so on. So that's what cell-mediated endocytosis is when the cell is triggered and then it does the engulfing because it's triggered from outside. And the reason, of course, vesicles are able to fuse and unfuse with the plasma membrane is because they're made of pretty much the same bilipid layer structure of 2 layers of phospholipids. Okay, right in the middle here we have to give examples of molecules which use each type of transport. Passive transport tends to involve using molecules that are small and don't have much charge, so they can sneak across the plasma membrane. It also involves molecules that are following the concentration gradient. Whereas active transport is any molecule that's got to work against the concentration gradient, or something that's particularly bulky, like maybe the insulin that's produced in your pancreatic cells. The only way for it to get out is through exocytosis. Okay, so I've written a couple of examples here. So we've got water, obviously osmosis is a passive process. We've got carbon dioxide and oxygen gas exchange are fabulous examples of things that just diffuse passively across various surfaces in our bodies, like in our lungs and in our capillaries. Alcohol is another good example. It's very small and sneaky and gets into cells easily. Now, active transport, in your nervous system, you have special pumps, potassium and sodium pumps, and they will push potassium and sodium ions to make a gradient, and that's how our nerve impulses work. And I've already mentioned hormones as an example of exocytosis. Okay, we're going to move a little bit into the second key knowledge, which is about cell compartmentalization and the organelles of the cell. What cell compartmentalization is, it's creating little compartments inside the cell. So, for example, the nucleus is a little compartment where the DNA is housed. The mitochondria is another compartment with some very special internal environments that allow some very important chemical reactions to happen. Likewise with the chloroplast. So basically, it's almost like instead of just living in a house with no walls and things are all over the place, it's the same way as in our homes we have dedicated rooms for certain things. Like we have a room for cooking, we have a room for sleeping, we have a room for bathing, and it just makes our lives a little bit more efficient and more organized. It's likewise with the cell and its compartments, which of course we call organelles. Okay, so we've talked about cell compartmentalization, but it's really important to realize that not every cell does it. Prokaryotic cells don't, and eukaryotic cells do. Now we've got a table down here where we've got to talk about prokaryotic versus eukaryotic. Let's start with the kingdom. Okay, so prokaryotes, it's pretty much the kingdoms of bacteria and archaeobacteria. Whereas eukaryotes are plants, fungi, animals, and protista. In terms of size, prokaryotes are far smaller than eukaryotes. So bacteria sit about 1 micrometer, whereas animal cells start at 10 micrometers, and plant cells can be, on average, a little bit larger. So prokaryotes, another important difference is that they don't have membrane-bound organelles, whereas eukaryotes do. Eukaryotes do the compartmentalization. That's why they can be a bit larger, because they're more organized. Now, having said that prokaryotes don't have membrane-bound organelles, that does not mean they have no organelles at all. They do have things like ribosomes. They don't require organelles. Okay, in terms of their chromatin, their nuclear material, their DNA, the prokaryote has a single chromosome, a single central chromosome, but it also has multiple plasmids. And remember, they become important when we're speaking about some of the gene transfer stuff later on in the curriculum. Whereas eukaryotes have multiple linear chromosomes, but be aware they also have DNA in the mitochondria and chloroplast, and they're circular, like in the bacteria, which gives a hint to their past. Okay, so let's go through the organelles quickly, and I'll highlight the ones that are important for protein synthesis. The first one that's important for protein synthesis is the nucleus. Of course, DNA is housed in the nucleus and ultimately controls all of the activities of the cell. This is also where RNA transcription happens, and that's the first step in making DNA. You have to copy the DNA into RNA so it can go out and be translated into amino acids. To do an illustration, you just somehow show the nuclear envelope and the chromatin or the DNA inside, whether it's its chromosomes or as its unwound form. Mitochondria, energy production from the oxidation of glucose. So, of course, it releases ATP. You can use the term ATP in your exams. You might draw that as sort of a jelly bean and show that it's got an internal folded membrane. We'll look at that in more detail later on. Ribosomes are important for proteins. They turn RNA into proteins by linking up those amino acids. Generally, you might draw it as just little dots in a cell. But if you were to draw it closer, it's actually got a small subunit and a large subunit which sort of wraps around it, and that's how the DNA feeds through. The Golgi apparatus, we normally draw it as a stack of dishes. They sort of look like dirty dishes because there's often little vesicles sort of budding off at the side there. But that's a common way to draw Golgi apparatus. Cell wall, you might just draw it as a pretty tough, rigid looking thing. That's about all you'd have to draw for that one. Before I go any further, I'd like to point out that the Golgi apparatus is kind of important for the processing of proteins. Okay, rough ER. Now this is where proteins go after they've been made by the ribosomes, and the rough ER can be identified because it often looks just like a bunch of folded membranes, but you'll often show the ribosomes on the surface just by drawing dots to show that it's the rough ER. The smooth ER looks very similar, but it doesn't have any stubble. You could get away with drawing something like that. And be aware that that's associated more with the production of lipids, whereas rough ER is quite important in the process of proteins. Now, your lysosomes, often we just draw it as a little vesicle, a little vacuole. Just be aware that it's full of enzymes. And the cytoskeleton, I'm actually not going to try and draw that because I'm going to end up making a mess. Be aware that it's got a lot of roles. It gives the cell its shape. It helps with signaling pathways. It's involved in endocytosis. It moves those chromosomes around during cellular division. Remember the spindle fibers and cytokinesis. It organizes the contents of cells in space. So, keeps the endoplasmic reticulum generally close to the nucleus and so on and things like that. And of course, it can also be cilia and flagellus. So probably the most common way you know of it as a flagella is in your sperm cell. Sometimes you see it on the outside of bacteria as like these little hairs, and they're the cilia. Okay, and that's Key Knowledge 1. I'll upload Key Knowledge 2 very shortly for you guys.